Hey guys, welcome to Dublin. We're here for ISCT 2026. My name's Alicia Didsbury and I'm from New Zealand. And I just really wanted to have the opportunity to say we have brought back our star, Patrick Hanley, the Patrick Hanley, back for ISCT TV.
That's how old I am.
That's how old you are, yeah. Welcome back, Patrick.
Yeah, thank you. It's great to be here again.
So we're going to do a little bit of a recap about what's happened today and what have been your best take-homes.
Yeah. So I'm going to preface this by saying I was here yesterday as well.
Yeah.
So I was here for the commercialization series, and I just want to touch on that. We were arguing, one of the things was ex vivo versus in vivo CAR T cells. And the reason I bring that up is because it was an exciting debate. I think it's very clear that there's...
It's a hot topic right now,
Yep.
yeah.
And there's promise, but I think everyone appreciates that right now we can control the drug product by manufacturing it ex vivo. Once it goes in vivo, you don't know what you're going to get. You don't know the quality of the T cells. So, stay tuned for that. I think the investment in the field, these $10 billion deals is probably good spotlight. But I don't think anybody is really ready to move and say that ex vivo is over.
So when I have people, especially in government, come to me and say, well, actually, we don't need to build a CAR T platform right now because these in vivo CAR Ts are, just on the horizon. Do you think I should say back to them then, actually, let's just wait and see, we should get going on this other thing first?
And there's so many other cell types that you can use as well. I don't think that ex vivo CAR is going to be the panacea for every disease type, autoimmunity, solid tumors, viral infections. We need to invest in all types of cell and gene therapies, in my opinion, and not just cell and gene therapy. It's like all advancements, right?
Yeah. Absolutely.
Yeah. But the reason I was excited about that today is because then Jacques Gallipo, right, outgoing former president,
yep, asked
me and Massimo Dominici what our thoughts were, whether it was going to become the standard of care in the next 5 years, right? So even IST leadership, I think Massimo and I both said, yeah, we're excited about it, but I think it's a little bit too early. Yeah. Also, there are incredible similarities to the allo boom from 2022.
Yes.
So we'll see. I do hope for patients that it works out, but still too early to tell. What else have you seen?
I went to a really interesting roundtable this morning, actually chaired by you. I just remember that. And just some of the discussions about best practice and how we can work together to really get these therapies out the door, but in a less centralised manner.
Yeah.
And that's really pertinent to where I'm coming from, where, we don't have a big industry at the moment. Potentially, we will never have a big industry. And we need to take these other opportunities, and it's amazing to see you guys especially trailblazing this, and we're watching it very closely to see if there's an opportunity for us to really imitate that and try for the same sort of technologies and the techniques that you're using essentially to make it work.
And so that session was building a better business model or sustainable business model.
Yeah. And I loved the idea that actually using pediatrics as a test case to get things going.
Because we work so well together,
It makes really good sense, and likewise our pediatric community is really well connected, so it's a good space to be in. Yeah,
I was really excited about that panel, and that group is outstanding. So
wow.
What's happening at Fondazionale Telethon?
Yes.
Amazing, right? This is a charity that was started by phone calls to raise money.
Yeah.
And they just had a commercialized gene therapy product for Wiskott Aldrich, right? Like, that's why I wanted to have that session and say, hey, does this need to be this bloated $10 billion company? Yeah. Or can we do it differently, leaner? It's happening in India with the ImmunoAct. They've treated 700 patients with this model.
Which is just mind-blowing, right?
Yeah, $30,000 a dose. So, how do we do this differently? I was hoping that they'd talk a little bit more about the business cases and business models.
Yeah, because those are the things that we actually need to do, right?
Yeah, but everyone always loves to talk about CMC and regulatory, right? And so, whatever people want to talk about, we'll talk about, but I did want to talk a little bit about the business and fundraising and things like that. Yeah. One of the things that I was excited about was our Cytotherapy Associate Editor meeting, because we heard from Ed Horowitz, the Editor-in-Chief, about the impact factor that's coming up. So it's not released yet. We don't know what it is, but we've kind of done some behind the scenes calculations. We think that it's going to be favorable for ISCT, which shows the commitment of the society to the journal Phototherapy.
That's fantastic. That's fantastic.
So no promises about what that bump's going to be or if there is a bump, but I think there is an increase in the quality of the journal. The turnaround time is better than it's ever been, and that's been a huge
super
focus.
fast, right? Yeah.
So
no, you guys have done an amazing job, and I love how you've extended the editorial team to include early career.
Yep.
That's been a real star thing to do, and I've had a lot of people come and say to me, "Should I get involved in this?" I'm like,
yes,
get involved. It's fantastic.
And congratulations, by the way, on the ESP Superstar Award.
Thank you, and congratulations on the. I was going to say middle-aged.
No, it is. It's the elderly
on
award.
The mid-career.
No, what I like about the award that I won is that it's the continuation of the work that you're doing, and it gives someone a stepping stone, right? So I'm grateful for the award, but I'm excited that it's available now for the next generation. That it's a continuation of what we started with the ESP committee in 2015. Right. So.
It's it's interesting to see the journey and seeing the ESP committee and the leaders coming out of it. Like I'm thinking about you and Kevin coming through and just doing so well. And it's an exciting space.
Yeah, Kevin's also great to work with. He's been leading this white paper we're writing on a new BLA path.
Oh yeah.
A recommendation for the FDA, and so stay tuned.
Superstars. Anything else you want to touch on? What are you looking forward to next? Tomorrow.
So tomorrow morning, nothing better to look forward to than a 7 a.m. champagne and citations.
7 a.m. Champagne though.
Well,
did you actually get champagne?
Well, we'll find out tomorrow whether we get champagne. But no, I think I'm looking forward to the 30 years of cell and gene therapy.
Yeah.
So we'll see what happens tomorrow.
That's fantastic.
Yeah, what about you?
There's a couple of amazing roundtables that I'm really looking forward to. One is actually around crossing borders and access across borders, particularly within the EU, and I'm really looking forward to seeing that. Yeah.
And I also wanted to close with a shout out to Cleo Rooney, who won the mentoring award, the Darwin Pop-Up Mentoring Award. She said that she wanted to watch the ISCT TV. So Cleo, congratulations on your award, and I hope you're watching this.
And I'm just going to say something which I was too scared to say to her. She's been I've been reading her papers for so long. She's such a superstar. So it was so amazing to be on the same stage as her.
No, she's a great person. And she's always kind of flown under the radar because there's so many other people at Baylor that, get a lot of the recognition, but she has so many great ideas, and she's really behind the scenes mentoring all these people. So it's great to see. By the way, former ISCT president, incoming ISCT president, Telegraph editor, myself, multiple chief scientific officers of biotech companies. Just an incredible legacy.
Yeah. Amazing. All right, guys, let's wrap it up for now, and we will catch you tomorrow.
See you tomorrow.
See you.
Welcome to ISCT TV, and my name is Mikey Crean, and I'm your host, and I'm here with the Roundtable Programme co-chairs, Emily Blitz, Dominic Clark, and Wade Atkins. Welcome to Dublin. Are you enjoying your time so far?
Oh, I love Dublin. Yeah.
So far, yes. You guys have turned on the weather in an amazing way.
That's it. I see you've brought the good weather with you. The sun today, all the way from Australia.
All the way from Australia. It was a very long way.
Great. So, the roundtable sessions today have been very, very busy, and super great attendance. While you're here, I'd love to get your insights on how the topics were picked and how you chose them this year for the roundtable program.
Well, the way that they're proposed is via all the standing subcommittees in ISCT, of which there are a large number covering a huge number of subject areas. So those subject experts in the committee put together a proposal, and then that's assessed. So we had such a huge response from all the subcommittees this year, and it was really challenging to cut it down to what would fit into the program. So we assessed them for a whole bunch of factors, but also the main one really is that it was something unresolved that needed discussion. So an issue that's current. Wade, you gave us a really good list in your introduction earlier. What was the list of criteria?
We wanted to make sure they were cutting edge, and as you said, unresolved issues. They fit with our theme of this is the golden age. We're coming to our own lots of approved or licensed products globally. And so they need to fit in with that and connect to how we're taking our industry partners and blending with the clinical side, with the lab to figure out solutions to advance to therapies for patients who are in desperate need. So it's really about open, honest discussion about solving the problem today for the patient that's waiting.
Yeah, and I think that's the critical part was on top of all that, we've got these unresolved challenges, and what we really wanted to cultivate beyond that is debate, right? We want not just talking to the delegates, but an actual interactive debate, because that's how it's going to move things forward, right, and solve these challenges that we continue to have and continue to make progress on.
And is there any particular of the topics you expect that's going to create the most debate that you've seen from your program, from the 25 roundtables, that you're anticipating, or even today, there was some of them that's caused some debate?
I'll start with one because I think it's easy in a way. AI, right? Everybody talks about AI, and when you really dive into it, we're just barely touching the surface. And, so where do we use it? How do we use it? Are we really using it? And so I think there was already a debate today on should we be using it? And I think ultimately, there was plenty of discussion. So for me, that was one of the most interesting ones so far.
And are we using it responsibly as well? That's exactly it.
And the discussions this year end up feeding into the topics of education for next year or the year after, or the decision making that gets made. I mean, the topics that are of great interest to me are the ones about patient access. So there's quite a few, we're a global society. There's, we are working to get patients access to effective and safe therapies all over the world. And so, there's issues of different jurisdictions. One of the really interesting topics that I went to this morning was about how do you balance that question of safety regulation with what is actually required but still allows the patient to have access. So there's going to be a sweet spot there, but how do we work out what the sweet spot is that's not going to be so prohibitively expensive to comply with? That's a really interesting topic, and that one is one that needs discussion. We need discussion between the inventors, the process people, industry, the clinicians and the regulators to find a solution to that one.
Absolutely. And there's a particular roundtable tomorrow. Turns out I'm actually the chair of it, is on cross-border access of approved and legitimate cell therapy. So if people are around, it'd be great to attend that. And Wade, I'd like to get your...
So I think we're all in our little silos and niches, but the ones that resonate with me the most are, I'm a little biased, around regulatory and quality in a global setting, because we have so many variables about how regulators do their job about are the products safe when we are a global society and some technologies are available in parts where patients desperately need them somewhere else. So how do we navigate the requirements to protect patients in a region while we're bringing life-saving therapies to very small numbers for rare diseases? So I think discussions about how we rethink regulatory and how we standardize quality using our basis for ICH documents like 5, 7, 9, and a global approach to solving humane problems. And I think sometimes we step back and think about our own situation, our own environment, and some are very privileged to have a lot of access. But to what Emily spoke about, how do we have access globally, safely, compliantly, and equitably? So if you're a patient in Singapore, are you getting a similar treatment as the US or in the UK or in Australia? So how do we solve that problem? It's very intriguing to me.
Yeah. I think that's just goes back to the strength of this community and the strength of the individuals that are coming together to submit the ideas for these roundtables, to ultimately have these discussions where it's not just each one of us, as Wade just described, we're having these individually, thinking about these challenges, but we don't make progress unless we all sit down in a room and converse about them. And that's where I think the roundtables fit right in with the rest of the program, because it all connects.
Yeah, and that totally fits with ISCT being a global society, getting everybody in the room to have these types of conversations, etc.
So a reflection in that theme, Dominic, as last year we had a roundtable about how in Spain they were having point of care testing and had broken down some of these barriers, trying to bring it local to the patient. And we heard that. But also there were members in the audience from the US Food and Drug Administration. And we've seen over the year how they're changing their approach to where this is possible. So it's that where if it works in one region, can it work in another region? And that open-minded thinking and coming to the table about solving the problem and breaking it down, identifying what the problem is, because I'm convinced in this society, we are smarter than the problem if we can define it.
Definitely. And before you finish, there's a fun fact trivia, Wayne, that you'd like to maybe give us some insight into?
Well, I was very piqued by Emily's comments about how we had a lot of submissions and how we had spent a lot of time with a committee vetting these. And I was reflecting that probably 10 to 15 years ago, I pitched an idea about having a session where we didn't have didactic slides. We just talked about the problem and how are you approaching this and how are you approaching this, and it morphed into round tables. So maybe all of this is my fault.
You heard that. Blame Wade.
Yeah, that's right. If you don't like it, blame Wade. If you do like it, that's me.
Yeah, if you like it, it's Emily.
Well, we are totally delighted that this roundtable sessions do happen. So thank you for all your time and effort, and also thank you for your time today for the interview.
Yeah, thanks, man.
Absolutely.
Thanks very much. Thank you.
Thank you.
Oh, there's Lise. Come on. We're going to interview her. Hi, Lise. How are you?
Oh, good. It's nice to see you.
Yeah, good. So it's your first ISCT this year.
Yes,
Have you enjoyed it?
It's very nice. It's very international. You have people from the US, but also from Asia, from Europe. And everybody's very enthusiastic and very open to help and give more information. So
Brilliant. And we had a great cross-border access roundtable today. Plenty of audience engagement, which was great.
yes, indeed. I think it was a very nice one. Happy to show the work that I have been doing. It was also complementary with the panelists. And indeed, there was much engagement from the audience, who was willing to share all the experience they have from US, from Australia. So yeah, it was really nice.
Well, thanks a million, Lise. I hope you enjoy the rest of the conference.
Thank you.
Talk to you soon.
Bye.
Okay, come on. Let's see who else we can find. Oh, there's Anna up here. Come on. Let's go.
Hi, Mike.
How are you?
Yeah,
You enjoying the conference?
Yes.
Yeah?
Yeah.
Any sessions in particular that's standing out to you?
Oh, actually, there are a lot of cool sessions, a lot of science, a lot of ITMPs, and a lot of progress. But it's not only science; it's also about mentorship. I really enjoyed actually your session.
Oh, cheers, thanks. Yeah, we had a good session. Yeah.
Yeah, it's give a perspectives. It's career one thing, but there are also I really like a quote yours that you need to absorb, and then also that you need to basically everybody here is smart, but you also need to be kind. It's really reflected, isn't it?
Thanks a million. Well, enjoy the rest of the conference.
Thanks.
And enjoy a few Guinness on me later, maybe.
Same.
Hello, everyone, and welcome to ISCT TV. I'm Elani Wiest, and today I have the honor to talk to both a singer-songwriter, Max McCarthy, and scientist Bruce Levine. Thank you guys for being here today to talk to us about your new song, Ring the Bell. So can you guys start off by just telling the audience how your paths crossed and what inspired this song?
Yeah, so basically, I remember my family was diagnosed with MDS, and I was doing a lot of research about CAR T-cell therapy, and I came across Bruce and we connected. I sent him an email. He's actually in Galway, and then I asked him could I meet up with him because I wanted to learn more about all this CAR T-cell therapy. And he said, yeah, I'll meet up with you. And we met in Dublin, which I was blown away by. I was just flabbergasted to see what they were doing in the field of cell therapy, and especially around CAR-T at the time, what they did for Emily Whitehead. And so I started getting a real interest about it and how it works. And of course, I have no better teacher than Bruce Levine. So, I'm learning every day from him. It's amazing.
Yeah. I mean, just picture this. It's the first meeting I go to after pandemic. It's still in pandemic, November 2021. I give my talk, I check my email. The subject line is Help for Cancer, hi Bruce from Mags. And I open it up, it says, I'm writing to you from Ireland, wanting to know about CAR T cells. Can we set up a Zoom? And I write back and say, actually, I'm in Ireland right now. So what are the odds that I was in Ireland of all places on that day?
Yeah, it was bizarre because he wrote back within minutes, and I was like, oh, he's in Galway? Oh, that's not too far away from me.
Well, it was obviously meant to be.
Well, that's it, you see. So
Bruce, you wrote some of the lyrics in the first version, if I understand correctly.
I had this crazy idea one day, what if Mags had her own song about CAR T cells? So I sent her some words just as a joke, right? And she wrote back, OMG, I have my own CAR T cell song.
Yeah.
And then about 20 minutes later came an a cappella version of one of the verses. I think the power of the song, what started out as kind of a lark, we purposely turned it into a patient story and a tribute to patients fighting cancer and the families that support them, and an effort to raise awareness of CAR T cell therapy and cell and gene therapies in
Such
general.
an amazing way to raise awareness.
absolutely.
And how do you think a song and a storytelling advances or advances or humanize the science?
Well, I think for me, when the song was done and the words there, but it didn't really, until the music video was put together. So Bruce had, reached out to certain patients that they were treated them, and we included them pictures of images in the video. And until you really see the video, it will break your heart. It's actually amazing. And I showed it to colleagues of mine, and you cannot watch that video and not cry. And so the lyrics are, I had all the scientific way to go with the song, and then I humanized it, so it's understanding from a perspective from my end. But it was really, for me, it was when the video came in on top of the song. It tells the whole story.
It's powerful.
It really does.
Yeah.
Very powerful.
Very powerful.
And I worked very closely with Max's producer on that video, and I asked permission from patients, would you share, here's the song, we're making a video, and place each one in the song at the lyrics that make the impact to convey the message. And I can't watch it today without even getting emotional yesterday when we showed it.
Yeah, absolutely, because as you all know, some patients don't make it through either, and that verse is there. And so I'm very grateful to all the families that did share them pictures, because it's very difficult. But it's telling the story, it's getting the word out there about, and it's getting hope. It's giving people hope. Every day we live for hope, and we live to survive. And if we can help somebody else, and music outside of cancer, music is very good for us anyway. Music, there's something touching about music, and I was very grateful to be part of this. I'm very grateful to be here and seeing all these amazing companies and all the amazing clinicians and scientists that I've met. It's really truly incredible.
So how can we create more platforms like this to bring more awareness to other diseases and other therapies and hope to those patients?
Well, more music. And we have to think about not just speaking amongst ourselves as scientists, but communicating to patients and the public about what we're doing yes, the technology behind it, but the work that we do is ultimately to benefit patients.
Knowledge is everything, yeah.
Ultimately, it's the patients and the public that pay for our work through taxes, investment, philanthropy. So we owe that debt to the patients and the families and the public.
Yeah, science communication is so important, and what a better way than communicating it to song to reach so many more people. So I'm really thankful for you guys for coming to talk with us today, and
thanks very much. And I'm glad it came to Ireland, ISCT. I was really excited, looking forward to seeing it today, and it blows my mind. Yeah, so thank you so much. Thank you, Bruce.
Yeah,
Thank you.
Ain't afraid of no cancer cell. These CAR-T warriors break the spell. We program to recognize the foe. Watching tumors wither and go. Guided missile, a CAR-T cell. Living drug to ring that bell. Ring that bell. Oh, guided missile, a CAR-T cell. Living drug to ring that bell.
Karen, we spoke last year about New Orleans and it would be coming to Dublin, and now we're here. It's great.
Amazing.
Yeah, it's great to have all these cell and gene therapy experts here. The sun is out and we have one of our experts here, Samia Viswanathan. Great to have you both. So, Karen, you chaired your session today on MSCs, and can you maybe elaborate about what some of the talks happened
Sure,
today?
absolutely. So I will say it followed on from Somia's excellent roundtable discussion yesterday, and I'll let her delve into that. But we followed up our conversation today, focusing on the interaction of MSCs and macrophages. We also had a little bit about neutrophils as well from one of our speakers, Nadir Kadri from Karolinska in Sweden. An excellent session. We had Somia speaking about her work, looking at the reprogramming of macrophages in OA. Really, really fascinating data. And, she told us about her exciting, work towards looking at maybe an epigenetic imprint in macrophages in the joint. We then heard from Tracy Hang, from Monash in Australia, who talked to us about really exciting data that she has found in the spleen in animals that receive MSCs. And it seems like the macrophages in the spleen become repolarized, and they're much better at using iron or processing iron, and that seems to be important for their therapeutic effect. So a really, really exciting session. And what I would say is, look, watch this space, and I think we're really going to see an innovation and hopefully more and more people coming together to work together to answer the important questions about the mechanism by which MSCs work.
Brilliant, brilliant. And Samia, there was a lot of talk yesterday about patient responsiveness, but today is about, as Karen mentioned, about MSC macrophage interactions.
Yeah.
And specifically about your talk. Can you just kind of tell us a bit more about that?
Yeah, I think the meeting's been really interesting and exciting, and we're getting kind of deeper into mechanisms of actions of MSCs, and I think that goes to address some of the heterogeneity questions, right? And why it's working in some patients and why it's not. But it seems that this interaction with immune cells, which is very patient specific, is what's driving some of those differences. So I think the two fields will converge, right? So what we learn from the mechanisms, we can take back, whether they're done in animal models or they're done, joint on a chip like we're doing, or in vitro cultures. We can learn from that and then translate that back into how maybe we need to start phenotyping these patients, immunophenotyping them, understanding their macrophage, their neutrophil, their spleen, right? And how they're processing these MSCs and why that's resulting in the different behaviors or responsiveness to MSCs that we're seeing in some of the clinical trials. So I think it's all a very full circle. But I think the depth is very exciting, and it's very much needed. But I think it'll circle back to informing clinical trials and clinical trial design.
Yeah, so that's brilliant, because there's so much innovative research going on in the MSC field now as well. But throughout the meeting, there's been so many other talks, and has there been any particular ones that really has caught your attention, or something that you're planning on going to today, or anything that's really grasped?
Yeah, so I've got a plenary that I'm hosting at 1:45 today.
Yeah.
Really looking forward to that, and that's focused on looking a little bit outside the box and maybe beyond CAR-T, and we've had some really really amazing, fascinating talks about CAR-T. But the session today, the plenary for Science and Innovation, will actually look towards slightly different versions of cell therapy that are doing really, interesting and exciting things. So those will be focused on macrophage-based cell therapy to target liver fibrosis, and that's by Stuart Forbes from Edinburgh. Really looking forward to that. We're also going to hear from Claudia Santos from Toronto in Canada. She's going to talk to us about her work on MSC and EVs in the area of sepsis even. And we also have Robin Parher, who's going to talk to us about CAR-NK cells, okay? And I guess the vision is that we're looking towards alternative cell therapies that can maybe address some of the pitfalls that are associated with CAR-T cells, such as toxicity and so on, and the fact that we are currently still focused on allo cell therapy for CAR-T, whereas these other indications can be allogeneic, that's auto for the CAR-T.
So so much to unpack and enjoy in them sessions. What about yourself?
I think I'm also, an MSc expert, so obviously looking forward to the MSc sessions. But tomorrow there's a lot of EV sessions, so MSc EVs are also very much at the forefront, so learning a little bit about that. About their mechanism of actions, engineered EVs, so I think that'll be great. But I really like the NAMS session that I was just in today, and it kind of follows really nicely from the keynote talk that Joseph Wu gave yesterday about NAMS and about clinical trials in a dish, right? And how we can maybe tackle some of the heterogeneity issues that we're seeing with patients using patient-specific organ on chips or, cells to maybe do the clinical trial or explore that heterogeneity or that interaction with macrophages in kind of models that are in a dish. So I'm very excited to hear about that today and yesterday. Yeah.
So much exciting things, yeah. So, thanks so much for your time today. I really appreciate it. So I'd like to say that it's been such a wonderful meeting here at ISCT, and I'm really looking forward to welcoming you all to Toronto next year.
Yes.
Yeah, brilliant.
Yes.